Hi, my name is Dominique Bergmann, and I'm a professor at Stanford in the Department of Biology, as well as an investigator at the Howard Hughes Medical Institution. And I'm going to talk to you today about stomata, so parts of plants, and plants' interaction with the environment. Part of this work has been done in collaboration with Joe Barry, who's at the Carnegie Institution. He's in the Department of Global Ecology. I'm actually associated with the Department of Plant Biology. But both of those are really essential institutions telling us a lot about basic science in the world. So this is part 3 of a 3-part series in which I've talked about plants, plant development, and stomata. And the first 2 were focused a little bit more on how plants grow and develop in the environment. This is going to be a little bit more about physiology and about climate change. So one of the key features about plants is that plants are taking carbon dioxide in from the atmosphere and bringing that into the biosphere, into the terrestrial life. And they're doing this through small pores on their surface called stomata. And those pores can open and close, and that brings carbon dioxide in, as you can see in the diagram. But when they're open, those pores are also letting water and oxygen out. And that's absolutely essential for a plant in order to be able to breathe. So plants are trying to get that carbon dioxide in. Losing water is not necessarily great for a plant. It dries out. If you forget to water your houseplants, you'll notice this. But actually water leaving through these stomata is normally an important part of a plant because this is the end of the transpiration stream. So plants live in the ground. There's water in the ground. In order to get that water throughout the plant, they don't have a circulatory system like we do that moves things all the way around. If they want water on the top of the plant, they actually have to bring it up from the roots up to the top. And the power that drives that is that transpiration, that evaporation of water through these pores. So they're very important for that. And also in the same way that sweat on our skin helps cool us, water evaporating from pores on plants helps to cool them. So these are very important pores, and they have a functional role for plants. Now, this is an ad that I saw a few weeks ago, and I just wanted to bring it up to say that sometimes people don't quite get what's important for a plant. So this is a plant, this is an ad that's supposed to be for something that you put on your skin to prevent dryness. And so as an example, this is Vaseline that you've put not on a leaf and then put on a leaf, and you can see where you've put Vaseline on the leaf, it's nice and shiny and hasn't lost any water. And that's essentially because this wax has been put over those stomata. So those pores now have a big waxy coating on them. And that's true that the plant would not be able to lose water, and that's going to stay greener. But it's also not going to be able to take in carbon dioxide. And so for this one isolated leaf that's going to die, that's fine. But normally if you put Vaseline on a plant, you'd suffocate it. Okay, so that's just saying that sometimes science should be left to scientists. All right, so plants are part of the world. They're a major part of the world. And this activity of stomata, this exchange of carbon dioxide from the atmosphere into the plant and water vapor back out of the plant, this is actually a big part of global climate cycles. So there's some numbers that I always find really fascinating. One is that if you imagine the entire water content of the atmosphere, so everything above us, that's getting recycled through stomata, those stomata that are on millions of plants over millions of hectares across the planet. So that's being recycled twice per year. So there's a massive amount of exchange between plants and the atmosphere. So plants are influencing the environment, but at the same time they're being influenced by the environment. And in the other parts of this, I talked a lot about how plants, their stem cells are controlled by elements in the environment. They make more or fewer stomata. They make bigger leaves or smaller leaves depending on the environment. And so this is kind of illustrated here in this yin-yang, where the activity of stomata influences the climate. What's in the climate comes back and influences whether plants have open or closed stomata, but also how many they make. And you can see this as an example. This is from an experiment done on a plant out in the desert. This is a plant that's a relative of potato. And this is an experiment where people change the carbon dioxide levels. So we talk a lot about how carbon dioxide levels have been rising steadily and they're accelerating. So one of the questions is, how do things respond? Now, if you think about a plant, plants are trying to get carbon dioxide in, and that might actually be an advantage for a while. But what does that higher carbon dioxide actually do to a plant? So in this plant, if you look at the surface of the leaf, This is at low levels of carbon dioxide, and this is at high levels. And at low levels, you see that there are not very many stomata, but each one of them is quite big. There are not very many cells in general. But when you make higher levels of carbon dioxide, the plants respond by making many more cells, many more of them stomata, but each one of them is smaller. And so there's this feedback back and forth. And when we want to talk about what's happening to climate and how we're modeling that, we need to understand how plants are responding. And so this brings up something that has been big in the news, and that is climate change, what direction. There's a lot of debate about it. People will see a graph such as this, and this is a graph that's been produced by the Intergovernmental Panel on Climate Change, the IPCC. And what they've been doing is taking data from many, many years and trying to predict what's going to happen. And this has huge implications for how, what we do as in terms of policy, whether we limit CO2, whether we make laws that will restrict certain activities. And so we really want to get this right. What you can see in this model, though, is there's a big difference between the first part of it. So in this part where we see these lines, this is actual data. So this is what's in the past. And this is a prediction of the future. And what you can see in this spread, even though you can't see the details, is that different models have different predictions. And so if we have really, really, really high levels, this is a temperature graph, we expect very, very rapid temperature rises. That is different than if we have only a modest one. And so we'd like these predictions to be as accurate as possible. So how do we do that? So one of the things that I'd like to argue then is that the more we know about the biology, the better our models can be. And the more we can come up with ways of testing small parts of those models, the better our models will be. A lot of times if you have a model that's trying to explain how the entire planet is behaving, we can't really do an experiment to test if that model is correct or not. So can we take parts of models and dissect them into something small enough that we can do a nice experiment? And with stomata, I think we have an opportunity to do that. So when we think about an overall climate model, and this is just an example from NOAA of what's going on between the oceans and the land and the atmosphere, plants are a big part of that. So plants are vegetation there and soils, and stomata are a big part of how the plants are behaving. So how do we do this? How do we find out? So if I've told you that plants change the number of stomata depending on the climate, and they open and close them depending on the climate, and they grow bigger or smaller depending on the climate, how do we get at this interchange? How do we maybe measure something about stomata that will help us understand how to make the models better? So one thing is that you can go around and go to interesting places and sample plants that have different stomatal properties. Desert plants don't have very many stomata. Jungle plants have a lot more. They're open a lot more. So one thing is you could look at a whole bunch of different plants and they have different stomatal properties. So that would be one way of testing how the Earth is responding. The other thing that you can do is take the same species, the same tree, that grows across a large range. And so this is an example of taking the same species of tree at different latitudes. So 38 degrees north is just north of San Francisco. 61 is Anchorage, Alaska. Look at the same pine tree in all of those different areas in between and see how that behaves. Or look at it at different elevations. Look at it at the top of the mountain or at sea level and see how they behave. And we can learn a lot from this. But there's a little bit of a problem in that if I look at these plants with different stomatal properties, the rest of the plant is really different. So this grass doesn't look anything like this pine tree. And if I want to know how this single thing behaves, how single stomata behaves, it's hard to uncouple that from all the other things that are different about those life cycles. A grass will only live for a year or two, a pine tree for hundreds. So all of those so there are a lot of differences between them. So we decided that experimentally, if we focus on one species that we could grow in the lab and we could control its environment very carefully, we might be able to provide a lot of information about how stomata respond. And if we could understand that in a very simple level, we could see if we can expand that out to see whether or not it would apply to other different kinds of plants. So the idea here is that you take a single plant and it's going to have different stomatal properties because through genetics and other experiments that we have in the lab, we've created or we've found plants that have different numbers of stomata. So in part 2, when I talked about different genes that we know about that make stomata or not so much make too many of them, But we can manipulate those, and we'll get plants that make fewer stomata or more stomata, or maybe put them in the wrong places. And so we can use that as a way of sampling, pretending as if we're looking at a whole lot of different plants with different stomatal numbers, but keeping them to the same plant. At the same time, we can grow that same plant in different environments, but we don't have to travel up and down the coast, the west coast of the United States. We can simply do this in a greenhouse, where we can change things like how much light does it get, the quality of light, that means is it bluer light or redder light, the temperature and carbon dioxide, et cetera. So by changing just a few things and seeing how stomata respond, we can get a much more accurate picture of how much stomata contribute to these global climate models. And I should back up and say, when we see those global climate models, and I just showed you an overall graph, each one of those has, among many, many calculations, there's always something that's trying to estimate how stomata are behaving. And the estimate is very rough, so we'd really like to improve that. Okay, so this is an example. This is using Arabidopsis, this small relative of broccoli that we do all of our experiments on. And we can actually, depending on what conditions we grow this in or using mutants, we can alter how many stomata plants make. I told you naturally they do that in response to different environments. We can also do that artificially. And so here's an example where we can change the density, so how many stomata per unit area. So on one side we have very few, and then moving over to very many. We can also look at the pattern of them, so we can have them next to each other, which isn't normally found in nature, or very spread out. Okay, so if we have, and so now if we can look at one species where we know everything else is the same, we're not trying to compare a tree to a grass, everything else about this is the same, we can isolate the effect of stomata in the whole process of responding to the climate. All right, so what we want to do is refine those models, and then the other thing we want to do is see if we can come up with a way, something that we can measure about a plant that's very easy, but it tells us about the rest of the health of the plant. So your blood pressure is something that's fairly easy to measure, and that's what that's supposed to do, is report on a lot of things, the state of your heart, among other things. So can we come up with something simple to measure that tells us a lot about the overall health of that plant? All right, so the thing that we really want to know is something called water use efficiency, and this is a key factor, a key feature for plants. So water use efficiency is essentially how much carbon you get in, so how much carbon you get in for food, at the cost of how much water. So the plant would prefer to get as much carbon as possible without losing water. And so that balance of water use efficiency is what we want to maximize. So how do we measure things, properties about stomata? So in part 2, I talked about making stomata. Here we're going to talk about the function of the actual stomata. So we need to generate something where we can change our environment in the lab, and that's this funny looking chamber up here. It looks a bit odd. We made it out of a fish tank and a number of other things, so it's handmade. And you can see the lights on there. Some are red and some are blue, and that's because plants need both red and blue light in order to survive. So this is actually, even though it looks a bit strange, it's actually a pretty good test of the normal environment outside. So we grow plants in those conditions, and then we use this machine. It's called a Lycor 6400. And what it does is it clamps down on the leaf, and it measures everything about that leaf. It actually passes gases through, so it pretends to be the atmosphere, and it asks, what does the plant take out of the atmosphere when it comes back around? So it's very accurate at measuring how much carbon dioxide the plant is taking up, how much water and oxygen and other things the plant is giving to the atmosphere. And so we're going to measure something called diffusive Gmax. What that is G is how much gas is going through the stomata. So it's called conductance. So carbon dioxide coming from the atmosphere into the stomata, the maximum amount, that's called Gmax. So we can measure the physiology there, and we can measure that Gmax at different environments. The other thing we're going to do is, after we clamp that leaf in that chamber and measure how effective it is at getting carbon out of the atmosphere, we're going to measure exactly how, on that same leaf, we're going to cut that out and count exactly how many stomata there are, how big they are, how deep the pore is that's underneath them. So a lot of things that we can measure pretty easily. But we want to see if we can make a perfect correlation between something easy to measure, like this, and something that we need an expensive machine to measure. Because if we can make those two things, so if this is the blood pressure and this is the heart, we'd like to make that correlation. So the key things to measure here are stomatal density, so how many stomata per unit area, how big are they, and are they next to each other in the wrong pattern, or are they spaced out? And that's something we call anatomical Gmax, so reflecting the fact that we're looking at structure, not function. So one of the first things we want to know is, well, if we just used these easy to measure things about how big the stomata are and where they are they good predictors of the physiology of the plant, how well the plant is functioning? And the straight line through the middle of the graph, if they're perfectly correlated, those two things, physiology and anatomy, that should be along this diagonal. And in fact, what you can see is that these control plants they're all along that diagonal. And when we started to look at all these different plants that we've made with more or fewer stomata, they also fall along that same diagonal. And so what that tells us is that we can do something simple like measuring how many stomata and how big they are instead of using an expensive machine to figure out how well they work. Okay, so that was actually done before, but there's a problem in that. And what those measurements are maximal conductance. So maximal conductance would be your stomata are wide open. What's the maximum possible amount of gas that can go through those? The problem is that plants don't usually live under those conditions. There are times when plants want to close their stomata. They don't want to be completely able to take in all the carbon dioxide that's available or lose all the water that they might lose. So in our current climate, we're not actually at that maximal conductance. We're at something below that. And those models that have been around before and our own models, that would be again this line, they don't really reflect the data, which is now down here, when we measure at a normal environment, the current environment outside. If we try to replicate that in our chamber, we're not very good. So what that tells us is that under certain conditions, measuring how many stomata there are and how big they are is a perfect measurement of how much the plant could possibly take in. But it's not a very good measurement about what it actually takes in today. So how do we improve that? So we use our mutants and we do a lot of other things and we start to calculate things. And essentially what we do is we run through a whole bunch of different environments. Down here at the bottom is something where we're measuring how much carbon dioxide is in the experimental setup. And these two stars, the first star around 400, that's our current carbon dioxide level, and the star around 700 is what we're predicting will happen in the next 50 or 100 years, depending on which model you look at. And so we wanted to know how plants with different numbers of stomata, the same kind of plant with more or fewer stomata, how that actually behaved under those different environmental situations. And so what we found, so this graph is color-coded. The blue bar at the top are plants with a lot of stomata, so their stomatal density is very high. We would guess that under maximal conditions, they would take in a lot of carbon. But we wondered what happened to those plants that normally have a lot of stomata under conditions where the plants don't actually want their stomata to be open. Can they close them all the way? That would be great. Or is there a defect? Under conditions where they don't want their stomata to be open, are they at a disadvantage? So we have those, and then we have a whole series of other ones, including some with very few stomata. And one of the ideas here is if we can find plants that behave well at our current climate, but also behave well at our predicted climates, we'll know in the future if we're trying to breed for things that will survive those future climates. We'll know what kind of characters to breed for. So if we look at this graph, and you look, so at one end is the maximal conductance. Things with a lot of stomata can take in a lot more carbon dioxide. That's not surprising. And those with very few take in very little. But if you look at the other end of the graph, what's interesting, this is a condition where plants don't want to have a lot of stomatal conductance. They want to be very closed. And what you see is that everyone kind of comes down to the same point. And so what that means is that if you have a lot of stomata, you're great under conditions where you want to have a lot open, but you're also fine under conditions where you don't want them to be open. So there's no cost to that. And that was a surprise, because normally in nature we see this balance, and yet if we interrupt that balance by creating plants that have more stomata or breeding for plants that have more stomata, they might actually do better under higher carbon dioxide regimes, which was a bit of a surprise. Okay, and this is just another thing. So another thing that we learned from this is regardless of how many stomata you have, it seems as if you can open them, which is going to let more things through, or close them equally efficiently. So if you have a lot of stomata, they're all open under certain conditions and they're all closed under other conditions. And this is just represented in a different kind of graph. And essentially what it means is that across the plant, if you have a lot of stomata, each one of them is responding to the environment kind of independently. They're not doing something as a group. And so what's important about that is that because each one is acting as itself, again, if we do something simple of measuring how many we have, that actually is a pretty accurate measurement of how the plant is going to respond in terms of how much carbon dioxide it'll take in and how much water it will lose. So one of the ways that we were able to make correlations between what you can see in stomata and how they function was using in a lab with some fancy equipment. But what we wanted to do, because we found that there was a great correlation, we wanted to come up with a way that nearly anyone could measure things about stomata, and that could be useful for contributing that to our global knowledge of how stomata behave in different plants. And so what we had was a microscope and some fancy things to look at this leaf in this way. And these stomata are very small. You can't see them without a microscope. But with something as simple as some nail polish that you can paint on the surface of the leaf and lift off and put that on the slide and then use your iPhone or something like that to act as a microscope, you can actually count the number of stomata on any kind of leaf. And so you're not restricted to something grown in the lab like we were, but you can actually go out and sample all sorts of different plants. You could sample those different plants at different times of the year, or in your neighbor's backyard versus yours, and you could learn a lot about them, something that's very cost effective. But if everyone does that in their own neighborhood across the US or across the world, we would actually know an awful lot about how plants are responding to the environment, particularly if we do this over time. And so adding that kind of information, how are plants changing their stomata, how is that affecting how plants are breathing and contributing to the climate, adding all of that into climate models is going to be really, really effective for learning a little bit more about how biology feeds into what's going on the planet. So this is work I mentioned at the beginning that has all been done in collaboration with Joe Barry at the Carnegie Institution. And Joe, you can probably see him maybe on the left, or you just see a bunch of trees. So he's out there sampling something at the top of the forest. But all of this work that I've talked about in terms of coming up with a way of measuring stomata and asking how their function and their anatomy fit together, it was all work that was done by a PhD student, Graham Dow, who's shown on the right with a watermelon. And all of that work was funded because Stanford University through BioX encourages interdisciplinary work. So people who work on genes and things in labs like my group, and people that work out in nature like Joe Barry, to try to come together and see if we can mix those two disciplines and come up with something really useful. Thank you.